Breast cancer is the second most common cancer-related cause of death for women throughout the globe. Breast cancer is metastatic cancer that can spread to other organs, including the bone, liver, lung, and brain, which is primarily accountable for its incurability. In India, breast cancer is one of the most common cancers, accounting for more than 27% of all cancer patients. Despite the fact that the exact cause of breast cancer is unclear, the associated risk factors have been discovered. Aging, a family history of breast cancer, particular alterations in the breast, genetic changes, menopausal history, lack of physical activity, alcohol abuse, obesity, diet, race, and chest radiation therapy are a few among the numerous risk factors that are associated with breast cancer. Recent studies have reported that owing to their cancer preventive potential, natural products will pave a way for more effective, non-toxic, non-endocrine, and therapeutic approaches for anti-cancer remedies. With modern technology and ideas, global pharmaceutical companies have started to rediscover medicinal plants as a possible source of novel anticarcinogenic drug candidates and have renewed their strategies favoring natural product-based drug discovery. During the last decade, we have witnessed a significant increase in the development and application of computational, in silico, methodologies in the field of drug discovery. Computational prediction tools have been widely employed to enhance the effectiveness of drug discovery because they allow researchers to test a large number of compounds in a short amount of time without incurring high costs or sacrificing animals. This can provide preliminary data for further in vitro and in vivo studies, which improves the study's success rate. In our study, we have employed a structure-based virtual screening approach to screen novel bioactive molecules from common Indian spices to identify potential inhibitors against certain breast cancer targets. We have chosen 5 Indian spice-yielding plants: Zingiber officinale, ginger, Cuminum cinnamon, cumin, Piper nigrum, black pepper, Curcuma longa, turmeric, and Allium sativum, garlic, as their broad-spectrum therapeutic properties are experimentally proved and well documented in various literature. From these plants, a total of 200 phytocompounds were screened against various breast cancer protein targets, i.e., cyclin-dependent kinase CDK8, progesterone receptor PR, and epidermal growth factor receptor EFGR. To determine the binding affinities of phytocompounds to 3 target proteins, docking studies were performed using the iGemDock 2.1 software. The results of our study indicate that Indian spice plants are rich in phytochemicals that can inhibit specific breast cancer targets. The top 4 drug candidates, namely apogetrin, cenerecide, curcumin, and xylaglucan, were selected. Among these 4 compounds, curcumin, apogetrin, and cenerecide were found to be the potential inhibitors of CDK8, PR, and EGFR, respectively. Curcumin has been demonstrated in several studies to have anti-cancer properties as it decreases cancer cell growth and metastasis by arresting cell cycle progression and apoptosis. Curcumin also prevents breast cancer stem cells, BCSC, an important factor that influences cancer recurrence, from proliferating. These experimental studies validate our in silico screening of potential inhibitors of breast cancer.